Let's review routine culture media and their purposes. Blood agar, also called BA or BAP, supports the growth of most bacteria. It is especially useful because it allows the observation of hemolytic reactions, which help differentiate species. Chocolate agar is enriched with hemoglobin, or iso-vital-AX, and it supports the growth of fastidious organisms such as Haemophilus and Neisseria species. Phenylethyl alcohol agar, or PEA, selects primarily for gram-positive cocci. It also supports the growth of anaerobic gram-negative bacilli by inhibiting other organisms. Columbia colistin nalidixic acid agar, or CNA, further selects for gram-positive cocci by suppressing gram-negative organisms. Thayer-Martin agar is highly selective for Neisseria gonorrhoeae and Neisseria meningititis. CAMPY blood agar is designed specifically to recover Campylobacter species, which require selective conditions. Thioglycolate broth is a versatile medium, often used as a backup for anaerobic organisms when primary recovery plates may fail. Lowenstein-Jensen agar is used for the isolation of Mycobacterium species, particularly Mycobacterium tuberculosis. Middlebrook 7H10 agar also supports Mycobacterium growth, offering an alternative to Lowenstein-Jensen. Finally, Petragnani agar is another medium used for the recovery of Mycobacterium species. When working with anaerobic organisms, special media are used to both recover and differentiate them. Bacteroides bile escalin agar, or BBE, selects for the Bacteroides fragilis group. Colonies that grow appear black due to escalin hydrolysis, making identification easier. Kanamycin-vancomycin-laked blood agar, or KVLB, supports the growth of Bacteroides species in Prevotella. This medium also enhances pigment production, which is an important feature for identifying certain anaerobes. Cycloserine C-foxidine fructose agar, or CCFA, is highly selective for Clostridioides difficile, the organism associated with antibiotic-associated colitis. Anaerobic PEA agar contains phenylethyl alcohol, which inhibits the growth of Enterobacteriaceae and helps prevent swarming of facultative anaerobes. This allows anaerobes to grow without being overrun by more aggressive bacteria. CDC anaerobic blood agar is enriched with hemin, cystine, and vitamin K, supporting the growth of a broad range of anaerobic organisms. Finally, cooked or chopped meat medium is a traditional enrichment medium that provides nutrients for the cultivation of a wide variety of anaerobes. It is particularly useful for maintaining viability in the laboratory. Certain organisms require specialized media for isolation and growth. Bordet-Gengou agar is the classic medium for Bordetella pertussis, the cause of whooping cough. Buffered charcoal yeast extract agar, or BCYE, is used to grow Legionella species, which require special nutrients found in this medium. Cystine-glucose agar supports the growth of Francisella tularensis, the agent of tularemia. Fletcher's medium is designed for the recovery of Leptospira species, which are delicate spirochetes. Sciro agar is selective for Helicobacter pylori, the bacterium associated with gastric ulcers and gastritis. Thiosulfate citrate bile salt sucrose agar, or TCBS, is used for the isolation of Vibrio species, including Vibrio cholerae. Vaginalis agar, also called V-agar, contains human blood and is designed to support the growth of Gardnerella vaginalis, a pathogen linked to bacterial vaginosis. Cystine tellurite blood agar, also called Tinsdale medium, is selective for Corynebacterium diphtheria. Colonies appear black due to tellurite reduction. Loeffler's medium is also used for Corynebacterium diphtheria. It enhances the grouping of organisms and promotes the appearance of metachromatic granules, which help with identification. Finally, CN agar, short for cefsulodin irgason novobiosin agar, is selective for Yersinia species and Aeromonas species. Biochemical reactions are critical tools in identifying bacteria. Let's review some common test media and their positive versus negative results. Carbohydrate fermentation with phenol red shows a negative result when the medium remains red. A positive reaction is indicated by a color change to yellow, reflecting acid production from sugar fermentation. Escalin hydrolysis is negative when no change occurs. A positive reaction is the appearance of a black color in the medium, indicating escalin breakdown. Hippurate hydrolysis shows no change when negative. A positive reaction produces a purple color, confirming hippurate breakdown. Motility testing is interpreted by growth patterns in a stab line. A negative result shows growth only along the stab line. A positive result shows diffuse or blurred growth spreading away from the stab line, demonstrating motility. 6.5% sodium chloride broth remains clear if negative. A positive reaction turns the broth turbid, showing bacterial tolerance to high salt concentrations. Phenylalanine or tryptophan deaminase tests are negative if no visible change occurs. A positive result produces a green surface when 10% ferric chloride is added, reflecting deamination activity. Finally, the urease test shows no visible change when negative. A positive result is a bright pink medium, caused by ammonia production raising the pH,